Yeah, was there right at the start of the development of spatial transcriptomics. His first method, seqfish, allowed sequential hybridization and imaging of a drastic increase in the throughput of imaging molecules within cells. And of course, using fluorescence in situ hybridization to look at spatial transcriptomics offers a huge benefit in that, as compared to the more conventionally done short read sequencing methods, there are no amplification biases, amongst many other things. Since then, he has been hard at work at improving the methodology, moving from expansion microscopy to the latest iteration where they utilize pseudo colors within the same channels to enable super resolution microscopy using a confocal microscope. He has many other developments that he's done, for example, memoir and many other things. Apologies. And today I'm very excited that he's going to be talking to us about how he utilized his modality to look at the DNA, mRNA, as well as compartments within the nucleus and reveal some pretty groovy biology. So I'll hand it over to you, Professor Kai, and we're really looking forward to your talk.
Okay, Sad. Thank you very much for the introduction. Let me share my slides. So, I attended this meeting over Zoom last year, and so I was really honored to be invited to give a talk this year, and I really hope to attend this meeting in person next year. So as I said, I want to tell you today about some recent work we've done where we look at nuclear structures, the architecture of nucleus in single cells, and by integrating both looking at RNA, DNA, as well as proteins in the same cell. And so just a quick disclosure, I'm a co-founder of Spatial Genomics. And so when we look in textbooks, we typically see that DNA, has this double helix, and then when we zoom out, we see that it's wrapped around histones. And then eventually, you get these, chromosome-looking like things that actually happens in metaphase. So most of the time, the chromosome doesn't look like this. And whenever you look into a microscope and you stain the cells with DAPI, you see that there are these complex nuclear stains. In the nucleus, you have these DAPI bright spots and DAPI dim things. And it's actually kind of hard for us to wrap our heads around, exactly how do you reconcile these two pictures, right? A very orderly structure versus something that looks very different among different cells. And, of course, the nucleus isn't made up of just DNA. In fact, there are many nuclear compartments, and these compartments are made up of a lot of times of proteins. So these things, we typically call them compartments or nuclear bodies, and so on. And so you can see, different structures. There are these nuclear speckles, and lamin, PML bodies, Cajal bodies, all these things together. And just highlighting that some recent work has shown that, if you look at the proteins, these are either transcription factors for mediators or transcriptional regulatory proteins. They seem to form globules, and there's some debates about whether these are phase-separated condensates. And so this is very active area of research, how proteins actually don't act as just monomers in the nucleus. It's not just a transcription factor binding to a site on the chromosome. It's rather they might form these globules that act together. So it's important to look at both the proteins as well as the DNAs. And I'm going to show you that just looking at individual components is actually leads to a very misleading picture of the nucleus. Whereas when you have all these measurements in the same cell, you can arrive at very clear interactions between the protein part and the DNA part, as well as the overall structure. It makes a lot more sense in single cells. So what people have done in the past decade in measuring DNA interactions and conformations in nucleus is developing these assays called the Hi-C, GAM, or SPRITE that measures interactions between pairs of loci or multiple loci at the same time. And from that, you can generate these contact maps of which loci is in contact with which other ones. And then through computational methods, you can reconstruct what the nucleus looks like. And of course, this requires a computational process, and usually this is done on bulk data. But most recently, there's been a lot of improvement. Technology allows us to scale this down to the single cell level, and this is work done by Takashi Nagano, when he's in Peter Fraser's lab, as well as his lab now, and also Sunny Shi's lab, where you can do these kind of Hi-C-like experiments in single cells. And on the other side, using the imaging approach, people traditionally have done, DNA fish, where they target specific loci for painting down chromosomes, right? But we want to be able to look at the genome at the level of, thousands of loci or potentially more. So how do we do that? I mean, one of the major limits in microscopy, is that you only have a few colors available. So on a typical confocal microscope, you have, let's say, 4 or 5 different colors you can uniquely distinguish. And here, you're actually looking at fish dots inside the cells. So you can convince yourself there's only 4 colors here. And the other problem is that when you have these dot-like structures in the cells, you put it on a microscope, you have this optical crowding issue that because each dot is diffraction limited, so you can only fit a fixed number of dots in the cell. So I want to convince you the solution to both problems is this sequential fish or SeqFish method that we developed actually for the last decade or so. And the idea is that you can target, in this case, just illustrated for messenger RNAs, but you can imagine this is done for all kinds of other molecules too, that you can hybridize against the same molecule multiple times. And with the imaging and stripping in between those hybridizations, such that, for example, you have this RNA1 here that is painted green the first time and then switches colors to blue and so on over the course of many hybridizations. And so this way, you can build up a barcode, a temporal barcode across multiple rounds, and it allows you to distinguish different species. So, if you start with 4 colors, just 4 rounds of hybridizations will give you hundreds of different barcodes and allows you to uniquely identify hundreds of different molecules. If you just do, twice as many, 8 rounds of hybridizations, you can already encode for the entire transcriptome or genome. So we showed that this can work at the transcriptome level, and this is by targeting intronic regions of RNAs. And so introns are typically expressed at the nascent transcription active sites, and then they're spliced out and degraded very quickly. So when you fish against the introns, what you're seeing is only the nascent site of synthesis. That's what you're seeing here is the nuclei, and each one of these dots is a single transcription active site that happens to be transcribing at that moment. And what we're doing is we're iterating through the different chromosomes here, and most of the times you can see 2 alleles. And then you have other nuclear structures like the nucleolus, as well as these DAPI-rich regions in the cell. And so in some sense, we got away with a little bit in this experiment because not all the genes are actively transcribing at any given moment. So out of the 10,000 genes, maybe only 1,000, 2,000 are bursting. And so you have few number of dots in the cells. So we showed that you can actually, if you want to apply this technology to look at messenger RNAs, which are much more highly expressed in cells, we're talking about hundreds of thousands or million transcripts total in a single cell. You have much fewer space to occupy to put all the fish dots in. So we showed that you can actually solve this problem using this method we call SeqFish Plus. I'll explain that in a second. But just showing you that in this cell here, we can zoom into this region and actually localize exactly where the individual transcripts are. And this is done for about 10,000 genes. And there's about 50,000 barcodes in just a single Z slice in the cell. So if you integrate over all the Z slices and then did more genes, you on average would get something like 500,000 transcripts in a cell. So how do we do this? So we use this method, SeqFish Plus. That's a way to do deterministic super resolution microscopy. And Sad mentioned that there are different other ways of using to do super resolution. One is using the traditional blinking, like palm and storm mechanisms, where you have a single fluorophore that blinks on, blinks off, and you can do super resolution that way. And the problem with that is that then you have to detect single dye molecules, right? Which is very, very hard to do in anything but a very flat cells. So if you want to do this in tissues where there's autofluorescence background, it's just like you can forget about it. And the other way is to do expansion microscopy. But then, if you expand the sample, you have to image longer. And also, one of the potential problem with the expansion is that the sample continues to morph during repeated rounds of hybridizations and makes it very hard to align the dots over multiple rounds. So the solution, we come up with is super resolution by coding scheme, which is that by using multiple rounds of hybridizations, you can target subsets of a very densely populated area. So what I'm showing here is that you have this region with many RNAs, and if you image them all together in a diffraction limited microscope, you will see that all these dots overlap with each other. But if you use sequential rounds Hive to call only a subset of them, and then you can Gaussian fit the dots to shrink their size and iterate through the different dots in this little cluster, then you can reconstruct a super resolved image, right, at much higher resolution where the dots are not overlapping. And you can repeat this over 3 or 4 rounds, and this way you can start to barcode the transcripts. So for example, this dot here showed up in Hive number 10 in pseudocolors, and on the first barcoding round, shows up in the second pseudocolors on the second barcoding round, and so on. So over the course of 4 barcoding rounds, you can decode exactly which gene this is because you designed the probe such that they spell out this particular barcode. And we did this in a way such that the color channels don't mix with each other, it decreases chromatic aberrations and so on. And this is how we can do this experiment with 10,000 genes. So you can think about this way of doing super resolution microscopy as, because you're controlling how the molecules turn on and off, right? It happens deterministically. And then each time a dot lights up, it's made up of tens of dye molecules because you have many dyes on this RNA molecule because you put many fish probes on it. So this way you increase your signal and you guarantee that you can detect things every time. So I think of this compared to the stochastic version of super resolution microscopy as a difference between American football versus soccer, right? Where you have very deterministic steps or something that's more like ongoing and random. So with this method, we can do not just cell cultures, we can also image 10,000 genes in tissues. So this is a piece of mouse brain slice. We can zoom into this region of the brain, mouse brain cortex, and zoom into further to the cell. And when you zoom in, you see this sort of pointillistic painting of these 10,000 different transcripts. There's actually many transcripts here is for this 10,000 unique genes. And then when you zoom in further, you can see all the locations of the transcripts with about 100 nanometer resolution in this experiment. So we can apply this approach now to looking at chromosomes in the cells. And we can do this in a way that, in the same cells, we can measure the RNA with RNA fish. So we look at about 70 different messenger RNAs as well as introns. And then we use the DNA-seq-fish plus to target thousands of loci. Particularly, we targeted, all the loci at 1 megabase resolution across the genome. So that's about 2,000 loci overall. And this is, I should say, this is done in mouse embryonic stem cells. I'll show you examples where we did this in the mouse brain as well. And then for each chromosome, we targeted about 60 regions. There are 25 kilobase apart. So this is a much higher resolution within each chromosome. And after all this is done, we can do immunofluorescence on about 17 antibodies for different epigenetic marks, as well as nuclear body marks in the same cells. So this way we can integrate all of these together. Okay, so just to show you some DNA-seq-fish plus data. So here you have a cell in one round of hybridizations. You can zoom in and you can see that you have these pseudocolors when you collapse all these different rounds together in this region, and they switch colors. So for example, this guy here switches the sequence. That tells you it's a loci on chromosome 10. And these guys, they switch in such a way that tells you they belongs to chromosome 4. So we're looking at this interchromosomal junction here. And this is for the 1 megabase data. And at the 25 kilobase resolution, we can walk along about 60 regions in parallel on 20 chromosomes. And then we can use a chromosome paint essentially to target individual chromosomes in the last 20 Hives. And that tells us that these guys belong to chromosome 15 and these guys belong to chromosome 9, right? And so we can basically reconstruct a very high resolution image of those particular chromosomes at those regions. And just to show you that data looks good, when we compare the seq-fish plus data with the Hi-C data, we see very good concordance both at the chromosome level. You can see the individual chromosomes clustered together here, and then at the 1 megabase resolution as well as the 25 kilobase resolution. And then so this way, I go back to these DAPI images, where we couldn't figure out exactly where the chromosomes are. Now we can go back and map out exactly where all 20 chromosomes at this 1 megabase resolution is matching to these particular cells. And so we can see actually there are many holes in the nuclear structure. I'll talk about that in a second. It's a really beautiful picture when you put it together. And you can, we got this data basically maybe a little bit before the pandemic. And so, sort of I sat there for half a year just staring at them. And if you just look at the chromosome structures, they're super beautiful. But the problem is that they're highly variable between different cells. So it's really hard to say anything meaningful about them, sort of as dimension reduction. Like how do you get something that's clear from these pictures? So this is where the antibodies come in. Like the protein actually allow us to pull this together. And so in the same experiment, we can stain against many antibodies, these different histone marks. And we do this by hybridizing oligos, the antibodies with oligos attached to them, and just essentially fishing them the same way we would perform the fish experiments. And this way, you can just use this on automated instrument and then go through them. And what we showed is that here we're looking at nuclear speckle staining here, that DNA fish is actually a very harsh step, that we developed this protocol where you fix the sample twice in a way such that even when you do DNA fish, that the immunofluorescence structures are not perturbed. So you can see that before and after heating, you have exactly the same structure. And so the problem with heating is that it reverses formaldehyde cross-linking, and this is what people do in chip experiments to dissociate the DNA from the protein. But if you fix it a second time with a different cross-linker, then even when you heat for DNA fish, then it releases the histone cross-link to the DNA, but it doesn't release the protein-protein cross-links. And this way you can preserve the nuclear structure very well in a way that you can align to the DNA. Okay, so this is what it looks like when you look at many, of these epigenetic marks. So the heterochromatic marks, the repressive marks, are sort of clustered together. You can see this in different cells here. The active marks, the K9 acetylation, the active polymerases, the nuclear speckles, they sort of overlap with each other, and so on. And you have this nuclear lamin, as well as again some heterochromatic as well as the active marks. And then finally, on the nucleolus, you can see that the chromosomes, these chromosomes in mouse, contain ribosomal DNA repeats, that they're associated with the nucleolus. So this is all validation to show you that you can align all these immunofluorescence images together with the DNA. And so this is the animation that shows that in the single cells, we're iterating through all the 1 megabase DNA loci that we decoded. And you can see in many cases, the chromosomes are separated into 2 separate alleles. And after the chromosomes are done, we're going to start showing the immunostainings. So these are the heterochromatic marks, the repressive marks, and they often appear in the same place. And now we're going to transition into this is the nucleolus, and now the active marks. So the active marks are a little bit more spread out, nuclear lamins, and then also repeat regions. So we can integrate all of this directly in a single cell. And what this allows us to do, right, then is to measure which DNA loci are close to which, chromatin marks, the histone modifications. And so we can generate, this DNA chromatin association map across the entire chromosome, across all 20 chromosomes for all these loci on 1 megabase resolution. So, for example, we see that many of these repeat elements, as well as repressive marks, tend to happen at the beginning, at the end of chromosomes, and this is known. The beginning and ends of mouse chromosomes are these telomeric regions that are repressed. And then if you look at this active marks H3K9 acetylation, if you zoom into this particular chromosome here, we can see that it has distinct peaks. And this is measuring essentially what's the fraction of time that each particular loci along this chromosome 11 is within 300 nanometers of the H3K9 acetylation antibody mark in the image. What you find is surprisingly that some of these peaks are associated with these active globules a very high fraction of the times, almost 95% of the times. And what's even surprising is if we compare this with the H3K9 isolation chip data, we see there's actually very good agreement between these two measurements, right? It's a little surprising because this is measuring molecular interactions, and this is measuring, spatial proximity based on imaging, based on, the DNA seq-fish plus is done in super resolution, but the immunofluorescence is done into fractional minute imaging. So it's about 300 nanometers. And of course, we're coarse-grained the chip data at the level of megabases, right? So there are actually more fine peaks in the chip measurement, but if you average all that intensity in a 1 megabase bin, you can see that they agree very well with the imaging data. And then, and this further point is that some of these loci are very consistently associated with a particular chromatin mark. So, you can see across the 400 cells when they measured in these experiments that many of them actually have the same loci that are associated with this particular mark. So this is not just specific to this particular epigenetic mark. If you look at nuclear speckles, you actually see the same behavior. So compare this now with the sprite measurement. There's very good agreement between imaging and sequencing-based measurements. And also you see these loci are highly deterministic at the single cell level. Now, because we got this from the imaging data, right, we can go back and look at where these loci are, right? We call them proximity points. And so that's showing these solid dots, whereas the protein globules are showing in these more transparent globules. So hopefully you can convince yourself that the nuclear speckle, which is these purple loci, the purple loci, the DNA proximity loci, they're situated in the exterior of these nuclear speckle blobs. And so that's what you can sort of see here, right? And it's the same for this heterochromatic marks. So H3K9 trimethylation.
You can see that. Settings.
Sorry, what's that? Yeah, so for the heterochromatic marks, you can see the same kind of the DNA loci are associated with the surfaces of these globules. And the same with nucleolus. And just to show you, this is not just the single cell, the cell 38 in this experiment here. If we look at different marks in different cells, again, these are the speckles and the repressive marks. But if you look at the other marks in the same cell, you see the same kind of features, that these proximity points, the loci are consistently associated at the 300 nanometer resolution with these globules. There all seems to be appearing on the surfaces of these globules. Okay, so it's different cell, same feature. And this is even true if you look at active polymerases. So if you target introns, and align them with the active polymerase mark, you can see that the introns surprisingly are at the edge of active polymerase globules. You kind of think that active transcribed loci are right in the middle of those things, but actually they also appear to be excluded from these protein globules. So I think the sort of the take-home message from this part is that when we typically think of these chip measurements, we think about the single track. You have these peaks along the chromosome, but in fact, many of these interactions may occur on these two-dimensional surfaces. And of course, if you go in and did super resolution imaging on the proteins themselves, then we might see even finer interactions. So what does these mean, right? What's the point of seeing these, loci, proximity loci, in single cells? Well, for individual chromosomes, you have these proximity points for different types of nuclear bodies. So for example, chromosome 4 has proximity points for heterochromatin at the beginning, and then for speckles at the end of the chromosomes. So in single cells, if you look at the cell here, you can see that chromosome 4 actually, sort of spans between these two nuclear bodies. And then in the middle, they're sort of jiggling around, doing things, but at the two ends, they're fixed. And same for chromosome 19, it has a lot of association with nucleolus, as well as the nuclear speckles. So you can see that this chromosome sort of straddles between these two nuclear bodies. It's just showing you a few other examples in different cells. You see this feature, this chromosome 4 again, spanning both the green heterochromatin as well as nuclear speckles. Same for the cell, you see two alleles. So this is for chromosome 17. You see the same features. Chromosome 19, again, for same cells. So the overall picture, right, is that these loci are sort of kind of consistently associated with nuclear bodies, right? Allows the chromosomes to be scaffolded across many different nuclear bodies. And if you look at just the chromosome itself, it looks very randomly organized in single cells, right? It's very hard to extract features out of that. But then if you just look at nuclear bodies, they also appear to be fairly randomly distributed in cells. But if you look at them together, what you see is that there's very deterministic structures in the cells, right? That along all these different chromosomes, there are points that has to be in contact with the nuclear body, or not has to be, but like very consistently associated with nuclear bodies, and they span different nuclear bodies. So this way, they sort of bridges the different structures in cells. And so what's something that's apparently random, if you look at individual components, when you look at both of these things together, it appears to be very, very deterministic. And this is just saying that, across the different 20 chromosomes, you have different marks where these loci are consistently associated with different epigenetic marks. But then, of course, we didn't do exhaustive staining of many chromatin marks. So I imagine that many of these white regions will get filled up as we target more and more epigenetic marks. And just to show you, right, this is everything I've shown you so far is done in mouse embryonic stem cells, but you can do the same kind of experiments in tissues. And this is done in a mouse brain, and we image these different regions in 3 different mouse brain slices. And we can use the RNA part now to identify the different cell types in the brain. So you can see the different, excitatory neuron, inhibitory neurons, excitatory neurons, and this is another type of inhibitory neuron, and so on, along with non-coding RNAs, DNA, and epigenetic marks all together in the same tissue. And from this, we can generate the same chromatin profiles for the different types of epigenetic marks. So this is for nuclear speckles, this is for K27 trimethylation. And some of them are very consistent. So for example, speckle association is fairly consistent between different cell types. K27 trimethylation is different between different cell types. And we see these features of these proximity points recapitulated in the brain, right? So in different cell types, in excitatory neurons versus astrocytes, you see these loci are consistently on the exteriors of nuclear globules. And what's interesting is that the interchromal structure, interaction structure is different in different cell types. So for example, chromosome 11 and 19 are highly interacting. They also interact with many other chromosomes in astrocytes, whereas a different set of chromosomes, 7 and 17, are interacting in the PV and excitatory neurons. And what gives rise to this structure is that chromosome 11 and 19 turns out to be close to the interior of the nucleus, right? And that makes more sense, right? Because when they're in the interior, they're sort of in contact with all the other chromosomes. And chromosome 7 and 17 are in the interior of these inhibitory neurons. And so we see actually a very good correlation between the radial score, so the lower the score, the more interior they are, with the interaction distance with other chromosomes. So chromosome, again, 11, 19 are in the interior, and they're also very close to other chromosomes on average. And why is this, right? So we can actually look at these proximity points and see if they can explain this. So if you look at chromosome 7 and 17, you see that they have a lot of interactions with heterochromatic globules. And in these inhibitory neurons, these heterochromatic globules, the green things, tend to be in the center of the nucleus. And so, these 17 and 7 chromosomes are then, by extension, tend to be in the interior. And the same kind of picture for astrocytes. It turns out that these K27 trimethylation marks, their globules are also in the interior in this particular astrocyte cell type. And that's why the chromosomes with those fixed points, the proximity points associated with that nuclear body, tends to be in the interior. This sort of reconciles the picture of, how do you explain that these are their overall chromosome features in the different cell types, And that can be partially explained by the fact that there are these associations, very consistent associations between DNA and particular chromatin marks. And one thing we found is that, when you look at the nuclear speckles, they're largely similar between different cell types, so there are subtle differences. And the loci that move closer to the speckles in excitatory neurons versus astrocytes tend to be more highly, expressed compared to the loci that didn't move. So you see that here, and versus the astrocytes that tend to be more repressed, in these excitatory neurons. Same for other nuclear bodies. So here for kind of repressive marks, they tend to be downregulated, in neurons and upregulated in astrocytes. So proximity to these nuclear bodies have relationships with expression levels. So it has a functional significance of how these loci are arranged in the nucleus. Okay, last point I just want to touch on is that if you look at just global levels of these epigenetic marks, the global levels differ in individual cells. So you can see, the K9 acetylation, this cell, right, has very different global staining intensity versus this other cell. And for different marks, it's different. And this is not a staining artifact. You can show that this is very reproducible. And in tissues in the brain, we see the same phenomenon. And in fact, these global differences in epigenetic modifications actually correlate with cell type on the RNA level. So you can cluster based on RNA markers, or you can cluster based on the epigenetic marks, and they seem to agree with each other. And on the single cell level in cell culture, we can actually measure how long these heterogeneities in the global epigenetic states last. And we can do a post-chase experiment by essentially measuring at different time points after cell division. And if an epigenetic marker is long-lived, then you should expect to see this colony to have very consistent features with a different colony with a completely different global state, but they're all coherent within the colony. But something that's interconverting between states very quickly will have a heterogeneous structure within the colony. And so for different types of epigenetic marks, we can see there are different features. Some of them are long-lived over, these two particularly actually are, have a memory of more than 3 cell cycles. And other marks like acetylation marks actually have very short memories. They usually are gone after 1 single cell cycle. We also see that chromosome structure themselves have 1 cell cycle memory, but I think this explanation for this is fairly simple. It's just that when the cells are dividing, the chromosomes segregate, and that's why within, if you just look immediately after that, the sister cells actually are very correlated in terms of their global chromosome organization. Okay, so let me just summarize and saying, I hope the takeaway message is that many non-repetitive DNA loci appear on the surface of nuclear bodies. So a lot of, maybe the picture of the nucleus isn't like a, spaghetti and meatball type of thing. It's more like inside a baguette. There are a lot of holes, and loci are actually appearing on the surface of those holes, but they're made up probably inside of a lot of proteins and also repetitive DNA, oftentimes in those nuclear bodies. And then when you put that together, you can see that the interactions between these DNA loci and the nuclear bodies actually imposes a very deterministic structure on the nucleus, and it's not really random. And it really severely constrains the diversity of the possible states the nucleus can exist in. And also there's long-lived states. So, I just want to end on a very broad point, which is that, when we do single cell biology, we oftentimes think about, how heterogeneous things are between different cells. But what we really want is to have dimension reduction, right? We want to say that's how we really understand something. That's what I would argue that's what insight is dimension reduction essentially. And this is important actually for many areas of biology. And I will make an analogy to this famous theorem in physics called Noether's theorem, and you can actually argue Noether's theorem is the basis of all modern physics. And the basic statement of this, one way to state this theorem is that there are things that are invariant. Like for example, there's time invariance. Laws of physics doesn't change when you look at it 10 minutes later. Also translational invariance, phase invariance. All of these things lead to particular conservation laws that give you equations. And in physics, I think this is the case because physics is low dimensional, right? At most it's maybe 10, 20 dimensions. But in biology, we have, 20,000 dimensions just looking at the RNA, and another 20,000 dimensions, you look at the proteins, all this other stuff, right? So we live in a very high dimensional space. We study something that lives in a very high dimensional space. So the more constraints we can put on, the more things that are invariant that we defined, right, the better. The more we're likely to arrive at something that's that tells us insights. And in biology, I will argue that looking for these invariants is as good as equations. We're not going to get to equations because there's just too many variables. Another way to visualize this is that, if you have these equivalence relationships, let's say you identify 0 with 2 pi, then, you can essentially convert something that's an infinite line into something that's more compact, like a circle. So that's the kind of thing I think that's really useful for us as a field, is to paradoxically that to arrive at these dimension reductions, we have to measure more things, right? And that's the value for doing single cell proteomics, as well as single cell genomics, RNA, DNA, and all these things all together. Okay, so the most important slide, I want to thank the team that did this, and particularly I want to highlight Udai, who's here, who did the DNA Sequential plus experiments, as well as Linus, who developed the RNA Sequential plus protocol initially, and as well as many members of the lab that supported this overall effort. And I want to also thank Barbara Wald, who was one of the first people that realized you can use sequencing to do functional genomics like ChIP-seq and RNA-seq. Previously, people thought, you do sequencing to sequence genomes, right? So I had this very random conversation with Barbara, almost a dozen years ago now, and that's what started us on this entire field of using single molecule microscopy to do genomics in single cells. So thank you for your attention, and I'm happy to take questions.
Thank you very much for that wonderful talk. Lots of information in there. So we have one question from the online audience, and that is, for seq-fish, after many rounds of stripping, will the signal decrease?
Yeah, so what happens is it's kind of funny. Your signal increases over the first few rounds of hybridization, and it slowly decays. And so for the seq-fish, RNA seq-fish plus experiment, after 80 rounds of hybridizations, the signal decreased by 20%. Right, so if you look at hybridization 81, which is exactly a repeat of hybridization 1, it's like 80%, 70-80% of the signal. And in between, you actually get more signal. And this probably has something to do with just loosening up the structures. Who knows, some black magic happens.
Thank you very much. Yeah, Professor Slavov.
Hey, Long, that was a beautiful talk. Really enjoyed it. And I very much appreciate the appeal of invariant relationships that we can find in the high dimensional data. I agree, that's the goal. So I have one question for follow-up work and one technical question. The technical one is, if you can give us a time scale of how long does it take you to acquire these data of combined immunofluorescence and DNA loci?
Yeah, so that experiment, the individual parts are done in a day or two. Like the DNA part is done in 2 days, the RNA part is done in 2 days, and the immunofluorescence is done in another 2 days. And so overall, it was like a week-long experiment.
For many hundreds of single cells that you analyzed.
In cell culture was 400, in tissues was thousands.
Great. And then I wonder about the possibility to correlate these structures that you see, whether within a particular cell type, between the different cells from that cell type, or between different cell types, if you can correlate these to transcriptional activity.
Yeah, so that's what I try to show a little bit. I think I went over that very quickly. Yes, so that's what we've done in this measurement here. So in the excitatory cells, you can see the loci are associated with nuclear speckles tend to be upregulated in terms of the expression. And then we get these expressions by taking single cell RNA-seq data and then averaging over all the cells of the same cell type. And you can see that, and you get the same for the astrocytes. The loci are close to astrocytes are downregulated in excitatory neurons.
I see. Very cool. And can this kind of analysis inform and contribute to the discussion whether DNA extrusion loops are part of influencing transcription? Do you have sufficient resolution for that?
No, so for this data, there isn't. So this is 1 megabase, and the extrusion loops happen on much finer time length scales, tens of hundreds of kilobases. So we can't say anything about extrusion loops here. That's a very good question.
Thank you.
We have another question here. Hi, thank you. Have you looked at the organization of genomically unstable cells like cancer cells of various different types?
Yeah, that would be quite interesting. No, we have not. And, obviously looking at the chromosome abnormalities would be very interesting to do. Arjun Raj has done these intron fish experiments. So Arjun's done them, I think almost a decade now. So the first people who did these intron fish measurements, and he saw, for example, in this cancer cell line that there's translocation that you can detect by just by even looking at the introns. The Philadelphia chromosome, I believe, was translocated between the two chromosomes they expect.
Okay, perfect. Thank you again very much for that wonderful talk. I think we'll move on to the next speaker.